Vora Baharov, and I'm a second-year postdoctoral fellow at the Eric and Wendy Schmidt Center. I'm delighted to kick off the afternoon session and introduce our next speaker, Dana Pe'el. So Dana is the chair of computational and systems biology at Sloan Kettering and is an HHMI investigator. Her lab's research combines advanced genomic approaches with various computational techniques, including machine learning, to address fundamental questions in biomedical science spanning gene regulation, cellular plasticity, and cell-cell communication. Dana received her PhD in computer science from the Hebrew University of Jerusalem, and she has numerous awards, including the ISCB Overton Prize in 2014. So with that, let's welcome Dana for her talk on decoding tissue architecture. Thanks so much.
And so really, things are fun. And this morning we saw super cool imaging technologies, and imaging is the name of the game. And there's lots of technologies, and we saw incredible work in cell biology. But I would actually argue that for my field of looking at tissues, and I call it painting molecules and adding hundreds of RNAs and where they are in this tissue and dozens of proteins, which is what we're able to do now. We still don't know how to interpret these tissues. We're actually not as advanced as my cell biology colleagues. Even something as simple as segmenting these cells is something that our spatial transcriptomics, haven't yet solved, let alone interpret this data. And one of the problems that we suffer, and this is why I'm a little bit jealous of my cell biology colleagues that we heard from in this morning, is that our problems are ill-defined. They come and, Jennifer said, go find me something that looks exactly like this ribosome structure that I've resolved, and that's what I want. It's a very well-defined problem. You can train an AI to do that, and you can tell an A to do that successfully. We, tissue biology people, are asking it, interpret this image. Teach me biology from it. And, AI is not magical, and we need to give it, in order to train it, we need to figure out what we want to ask it, and we need to give it the right training data in order to be able to ask it. So that's where my field is at. And, we've made, I'll tell you a little bit progress that we made. We need new algorithms. We need new foundations. We cannot take ChatGPT out of the box and say, let's throw it at this as well. It just won't help us to get new biology. And if you're in this audience and want to do anything meaningful, your goal should be, I want to use my toolkit, my AI toolkit, to really advance our understanding of biology and help people in medicine, rather than just say, hey, I'm doing AI. And so, here's a typical image. This is a slice of pre-malignant pancreas tissue measured with 480 molecules from xenium. And as I said, we can't segment it. So this is a really quick thing of an algorithm where we've made a tiny progress. It's not perfect, but it's benchmarked as the best. And here's a ground truth that we collected. Here's what 10x gives you out of the box. All the orange molecules are contaminations. These are molecules from completely different cells. If you include these molecules in your segmented issues, you're going to get completely wrong biology. And you're like, okay, why do you trust 10x? But Bezier, nothing else does better. We've benchmarked everything, and here's how you get all the wrong things. So there's just so many problems with the problem of associating transcripts to the right cell, even if you have perfect cell membrane segmentation. And so we just build a heterogeneous graph of saying these transcripts are next to each other, and these transcripts are next to a nucleus. And we're going to learn a latent of cells as represented by their nuclei and transcripts, so that the transcripts are near and correct to their right cell. That's what we train it to do. And we train it using the more correct transcripts on the nucleus. They're not perfect. We say, okay, those that align with the nucleus probably belong to that cell. Those that are in a nearby nucleus probably don't. We train it that way. And now we do link prediction. And lo and behold, it's not perfect. And we have this is still work in progress, even though it's available. And you can see we do a much better job, and we outperform on metrics and get that sweet spot of not getting contaminations. The solution of just looking at the nuclear transcripts isn't good enough, but also not getting incorrect or infrequently getting incorrect transcripts from other cells to get much cleaner biology for all the downstream analysis that we want to do. So now we have this image, which now we have everything linked spatially. We can assign the transcripts correctly to the right cell and map it back to the phenotypic landscape that we're used to. And now we can have accurate cell phenotypes embedded in space. Yay, that's a well-defined problem, so I can solve it. But how does one describe and interpret this image? You know, I was like, no, even, you know, let's define some questions. What type of niches, what type of environments exist in this cell? How to change when I perturb it towards malignancy under drug? You know, the problem is that, what do you mean? What is a niche? You have all these nice spots. I need a formalism for a niche. The reason single cell genomics worked so well is that we had a wonderful representation for cells. It's a vector of the expression of everything we've measured in that cell. And we had a semi-okay workable definition for similarity. You know, two cells are similar if the L2 norm of their expression is similar. And we don't have that for niches, and that's a problem. So what I'm going to try and tell you today in a pretty more technical talk, which I've gutted most of the biology out, is how do we begin to try and tackle that? And so the critical thing to get anything to work is we need to be, if we want the AI to help us, AI is actually really dumb. It doesn't know biology. It's not smarter than us. We can't just give it images and saying, you know, we can't figure it out, so you do. We need to tell them, we need to teach it what's meaningful. And specifically, what is biologically meaningful similarity between niches? And that, you know, is actually, you know, stumping the field now for about 2 years, because defining which two niches, you know, you just have these little snippets of images and tell me if this little field of view of a cell and it's like 10, 20 neighbors, and this little snippet of another cell and its 20 neighbors, is it similar or different? Is it meaningful? How similar and how different? Even something like that we haven't defined. This is work mostly by Doron, who recently graduated from my lab. He celebrated his defense 2 weeks ago. And a lot of innovation is thanks to him and not to me, but I'm going to tell you his story. So again, we're going to start with something fairly concrete. We're going to take an anchor cell at the middle, and we're going to look at its neighbors. You have to decide how many neighbors. Do you do it by radius? What resolution? I'm not even going to go into that, and I would say that different resolutions give you different biology. So if you ask me how big is the niche, I ask you, what is your biological question? And we're saying that a niche is a distribution. So we're going to try and treat the anchor cell and its behavior and its neighbors as a distribution. And actually, this is what the field has been doing without even knowing. Most of the field is collapsing this information, this very rich information, to frequencies of cell types. This is remnants from when we had 30 markers and all they could do is distinguish cell types. So if you have 30 markers to distinguish cell types, then collapsing it and saying, okay, I have 3 T cells, 2 myeloid cells, 2 fibroblasts, and 3 epithelial cells, that's all you can barely do, and that's all you can get. But if you have 480 transcripts representing rich states and rich differences between cells, which you've spent a lot of money to collect, collapsing it and throwing most of the information out the window is the wrong idea. So we want to keep as much information as possible in our distributions. So again, when are two matrices similar? Here are two matrices of cells and their neighbors. You know, some of you know this because I've been doing this for a while, but those of you who don't, are these two environments very similar, very different, sort of different? Who thinks that they're very different? Who thinks that they're, you know, they're intermediate different? Okay, who thinks that they're very similar? Most people don't want to vote. They're identical. It's a permutation. And you can get this permutation by rotating the image. I don't want the way that the technician put the slide into my microscope to make the whole thing differently. So we need a rich representation that's permutation invariant, and we need it to really not chuck away most of the gene expression I worked so hard and paid so much money to collect. And again, when you just take all the gene expression. I mean, this is a niche. This is permutation invariant. It looks at all the expression. Mean, that's been around for a while now. Recently, there's a couple papers saying, okay, let's take the mean of the niche. But you can see these are actually genuinely very, very different. They're not permutations, and we get the same mean. So we really have to stick to the full distribution. So we started by working, and this again we did a few years ago, taking one of my favorite tools, optical transport. There's a couple people here who love it. Optical transport is a very rich and expressive language, and you can basically say, okay, we want to find the permutation that makes this most similar. That's really, really nasty computation. But we don't like permutations. We want to smooth things. Let's just look for a mapping, a probabilistic mapping that's smooth, a soft assignment that says, let me try and basically move one distribution to the next. And we were really happy for it for a while, and then discovered, as much as I love optimal transport, I have data sets. A single postdoc now is collecting data sets of 15 million cells. A data set of 50K cells takes 2 weeks on my most powerful GPU to compute. That is not scalable. So we were stuck for a while, like, how do we handle these big data sets with optimal transport? I want to preserve a rich metric that actually is rich and biologically meaningful, and I want to run it on my 15 million cells and counting, and we just saw data sets of 200 million cells, and it's just going to grow. So again, each of these niches is a point cloud. It's a distribution of distributions. And so how do we model this distribution of distributions? And again, I'm going to give the credit where it's due. This was entirely Doron's aha moment. He got this aha moment when he was giving Journal Club on a paper that was using a transformer. This is why actually giving Journal Club can lead you to great insights by understanding other people's ideas and being forced to explain it to the group. Something clicks in your mind, and it's like, yes, this is exactly what we need. A transformer is permutation invariant. The fact we actually, for ChatGPT, force it and tell this is the order of the words. The transformer itself actually is permutation invariant. And it's, you could take different niche sizes. It doesn't need a standard input. So we have this transformer, and we're going to take our optimal transport distance and use a transformer to learn an embedding that actually is an approximation to the optimal transport distance in L2. So what we're really going to take is really small batches that we're going to choose, and for each of these batches, we're going to learn a latent and use optimal transport distance in the training to say, we want the distance of these cells in the latent space to match their optimal transport distance. We also want to be able to decode, so we want the optimal transport distance between the original input and the decoded input to be close according to this optimal transport distance. And yes, it's a little bit heavy to compute, but because we need to train it on just small batches, then I have this magical latent that then allows me, this is training. I can train very quickly in these small batches, and the latent actually works. So once I've trained it on some pairs and I have a latent, and if it works, then I can now take any pair of cells and compute their distance in L2, which is very quick. And if the latent is good, it will match optimal transport distance. And again, we actually ran this. We ran a ground truth for a month, trained on a very small subset, and actually got incredible accuracy. And I sometimes like to ask, well, how well did we do and how well can we do? And what makes me happy about this is there's some theoretical guarantees, and I can actually look at my data and know if I'm doing well or not. Now, this is all published in arXiv and in ICML proceedings. So, just to summarize very quickly, we can be accurate if our distance metric, we can embed our distance metric if our distance metric is Euclidean. And unfortunately, a metric is Euclidean if all its eigenvalues are positive, and optimal transport is not Euclidean. So this is certainly not Wasserstein distance. So it is impossible to get a perfect embedding. So I asked, okay, how bad can we go? Because, perfect is the enemy of the good. And what really made me happy is that, you can get really good, or the way you sort of break it is by having a lot of negative eigenvalues with heavy absolute value. So if your actual signal, your real positive eigenvalues, are really big relative to the sum of these small eigenvalues down here at the bottom, then you have a really good chance of doing really, really well. And this is fortunately the case of most real-world data sets that we've looked at. So now we have this latent space that approximates Wasserstein distance, but it is fully L2. And we can do all the fun stuff that we like to do with all the power and the ease and efficiency of L2. And I'm going to actually bring airplanes because no one's going to actually see cells and say, oh yeah, that makes sense. And even computing the barycenter in optimal transport space is really hard because you have to do all the pairwise things and stuff. But computing the mean airplane in Euclidean space is awesome. And another thing that we really like to do, and we saw actually a lot of examples today in the morning of actually something that we do in latents, is interpolation. We can again do these linear interpolations and take this length and turn it into a toilet gradually in this L2 space. And it actually learns paths because our latent space is a good approximation of Wasserstein distance. A little bit of biology. I said I gutted most of the biology and the really cool cancer stuff out, but neurobiology is very structured, so it's very quick to go through. And here we have Wasserstein applied to this neural cortex data. This is the UMAP data, and now we can actually say, let me actually define archetypical niches of this data set around these interneurons, which are sort of really small and they're dispersed and they have very different niches. And we can actually say, okay, find me inside the latent. Now what we're looking at is diffusion component in the latent that's approximating the wormhole space. And okay, what does this look like? And we see that this latent in the wormhole space perfectly recapitulates cortical depth of these neurons. Here's the diffusion component, here is the cortical depth mapped back onto the original image. Now we can look for these archetypical niches, looking at barycenters, and see how things change around in terms of neighbors as you go up through the cortical depth. And you can see that things shift around, the neighbors shift around. And even within a cell type, let's look at the blue cell types, these neurons right here. Here's how this cell type looks at this depth. And you go a little bit deeper, and you see that the same cell type is expressing genes in a completely different way. So while the cell type label has just collapsed this information, and if we were using just cell type, just the blue, then you would have thought it's the same thing. But look at how the state of these cells is so very different in these different cortical depths. And this sort of gives us these archetypical behaviors of this is the typical niche that you see at different cortical depths in the neuron. Now I said interpolation, so I'm gonna, Doran tried to push this forward and I actually thought it would never work. I told him, you're crazy. And so he's saying, okay, so let's actually try and take a cell and predict its environment. Generative AI. And so we're gonna use diffusion, flow matching. And basically you have data, it's hard to generate a sample from that. So you're going to sample from noise and learn to go from noise to a sample. That's what diffusion does. You have a forward process to learn, a reverse process, and what you really need to make this work is interpolation. But I've just shown you that I can do interpolation in this Wasserstein space. So I could take the classic flow matching algorithms, I can sort of lift them into the Wasserstein space through the wormhole embedding, which now allows me to approximate this weird Wasserstein space, but in a Euclidean world, and now go from noise to airplanes in this sort of space which allows me to map entire distribution. So this space doesn't map single objects, which works in Euclidean space. It actually allows me to map distributions in a meaningful way, actually using the Wasserstein optimal transport distance between these distributions in an embedding that approximates the Wasserstein space. So we can say, okay, how does it look like on these niches I just showed you? And this is like going from noise to creating, niches that look like real niches from real data. This is entirely generated by flow matching. And these niches actually really match the examples I showed you earlier, except they're generated from random noise to the beginning. And if actually you look at the real neurons and their environments versus those that I, pulled out and generated from, generative AI, you can see that it actually does a pretty good job. And, I was shocked because I actually thought it would never work. I'm a pessimist. So why does it work? It's the scale of the data. We're learning from, millions of samples. Each niche is very, very different. We're actually sampling a lot of biological variability. These niches are very, very different from one another. This is actually very different than some of the perturbation challenges you said at the beginning, where your perturbations challenges are, learn this thing after we've sampled a small number of samples that only take a fraction of the space. Here we have a very large number of samples that seem to capture the space, which is because some of the things I can say is that these models don't know biology, and a lot of the problem with these models, and everyone that uses them needs to be careful, is that they are still pretty bad at out-of-sample distributions. Anytime you take one of these algorithms that work and you say, look at how awesome it does, and look at my R squared, I ask you, show me it on out-of-sample and I'll believe you. Here we cheated because all these things I showed you wasn't out-of-sample. So as long as I'm not out-of-sample, it hasn't learned anything new. And we're working to try and teach it to learn something new now that at least we gave it a metric of real biology. But I want to, and again, this is something that I want to warn you. Foundation models are great, except for the fact that the real ones trained on trillions of images. And we don't have enough images yet to learn out of, enough biology to learn out of sample. And so where you want to be, in my opinion, the sweet spot for having impact is, yes, you can try and build, this is going to be a foundation model in the future once we have, 20 billion patients where we've thrown the kitchen sink at it. But by then, the AI architectures are going to be really different. So my advice to you is work where you can have actual impact today on learning something new, learning new biology, that, learning, having a real impact on medicine. And that's currently not the foundational models. Simpler architectures are, maybe less sexy, but they actually have enough data to give us real progress in biology today. What I really like and what personally excites me about this transformer that takes optimal transport and embeds it into L2 is you could use any easily computable optimal transport distance. This actually gives us a language where we can actually give rich, biologically meaningful distance metrics between biological entities and try and map them into a latent that now has more biology imbued into it. So just to give you a quick example, something completely different. We saw some AttackSeek this morning. I'm going to go through this real quickly, I promise. I'm going to now model distributions of cut sites. So this is AttackSeek data, and rather than taking peaks and whatnot, I'm going to ask exactly what is the distribution of each and every cut site. And we see that, different places we have very, very different distributions of cut sites. This is Earth mover's distance. This is the simplest of optimal transport distances that you have. And now we can take all these attack regions. We can take a region. We could look at their cut sites and define the optimal transport, Earth mover distance in this case, distances between all these little snippets of DNA profiled with AttackSeek. And, we try and push it a little forward. So this is the actual chromatin accessibility data. You see it has a really, really rich shape, and we were like really excited at first. And then we read Anschul's paper, and we realized most of it is actually TN5 bias. So of course, if you have that, it's going to create a lot of structure for any random sequence of DNA, irregardless of the cut sites. So what we really want is the signal beyond this TN5 bias. And this TN5 bias isn't exactly aligned, so again, we use Anschul's approach to actually learn a function of this bias using a simple net. Again, like classic nets that existed a decade ago, works perfectly. And now we're going to take the signal, we're going to remove the bias, and learn an optimal transport distance between the residual, which is what we care about. And now we have embeddings, and we can actually, ATAC is really sparse, so we can aggregate these embeddings in different ways. Here we're aggregating it at level of transcription factor in a metacell. This is all the places where we have CTCF motifs in a particular cell state during hematopoiesis. And we see that this has a lot of structures. The cells are colored by cell type. And we see at different cell types and different progressions in hematopoiesis, we have very, very different cut site distributions for CTCF, which again, both told us how important it is, how the genome and 3D genome is being completely shuffled, jiggered, and jigged over the course of hematopoiesis. We can take another viewpoint and say, let's take a specific region and look at how it changes across cells. So now we're going to take these enhancers, we're going to average them, we're going to pseudo-bulk, but we're going to look at the enhancer level, and we can see that there are very different regions that actually now, some of them are TF bound by CTCF, or CTCF bound, some of them are repressed, and some of them are active. And we can look and see that this structure has real meaning. We can find binding of CTCF with additional factors, CTCF with additional chrome modifications. These cut sites distributions have a very rich biology so that we can extract very rich biological interpretation from this latent that we just defined. In fact, we can even go towards a universal chromatin expression. So people like to go from, sequence to expression, from promoter accessibility to expression, and there's lots of models that do that really, really well. And we can use our latent to do the exact same thing, and it's sort of almost as good as these really high-end models that are designed to predict sequence from accessibility. Except what is the difference between this latent that we learned from the cut sites themselves is, these are very gene specific. So if you actually use this model on all genes rather than the individual gene it trained for, it collapses to random noise. We can train it on a subset of genes and then run it on test genes, and we've actually learned something real about accessibility that generalizes beyond individual cut sites, a real example of out of sample, but this is possible because we imbued our model with some biological meaning and a distance metric that has some biological meaning. So, what I hope you take out of this, beyond the fact that you should be, working on fundamental things that actually push biology forward rather than the fanciest architecture, is that I'm hoping that this tool allows you to take your favorite problem, use the awesome, expressive, powerful, flexible language of optimal transport to define what is a meaningful biological distance between these entities. And then you could use the wormhole transformer to embed it into L2 and explore to your heart's desires. We're using it right now as a building block of a lot of more fancy models to get a lot better interpretations of images, to distinguish between cell intrinsic, cell extrinsic, predict perturbations. But this building block of a biologically imbued latent, which actually has more biologically meaningful distance metrics, is something that I think really we need this and more things like it to push the field forward. I still have money, so there are positions, including staff positions for those who don't want to enter the academia right now. My single cell innovation lab also has lots of philanthropic money, and we're hiring. But really what's awesome is my group. This is Dorum. He did most of what I told you about, and thank you very much.
probably have time for one question.
Oh yeah, now you're getting back at me for saying foundation models are bad.
Not at all. Great talk, and it's fantastic. Now, my question is more about the starting point to define the niches. You say I represent a niche by a cloud point, a set of points, and you seem to forget about the relative positions of the cells. And I wonder if this is on purpose, or if you think a next version could be optimal transport on not only the values of the gene expression, but as well the relative positions of the neighboring cells, because in a sense, in your current interpretation, if I understand correctly, you don't care about how they are organized.
We've played a little bit with representation because, again, I've smoothed out and this doesn't work perfectly. So it helps get biology, but this is still work in progress in terms of getting the rich biology I hoped it would give. The exact order usually the exact order of the cells around it, their distances, don't impact things very much. The thing that actually makes a really, really big difference is how to actually represent these cells. So when we are at these data sets which have 500 genes, let alone some of these, Cosmex 5,000 gene data sets, actually this thing, optimal transport doesn't work really, really well, to go from such high dimension. And so actually featurization of the data, and how do I go and not represent all 480 genes, but actually represent them down to 100 genes. And again, PCA is too blunt. So basically getting better featurizations that aren't the full gene expression and not PCA as input is the way that we've seen the biggest impact. And we're trying a bunch of featurizations depending on the biological question that really help. That is where we need more, because we've pushed, and I already know that even the featurizations I've pulled out of the hat are not the best ones. So how to featurize the space is where the money is right now. But yes, optimal transport allows you to do all sorts of stuff. It allows you to go multimodal. It allows you to represent the geometry of the cells. It allows you to represent the distances. It's such an expressive language.
Okay, let's thank the speaker one more time then.